Welcome to our Johns Hopkins Grand Rounds for our Biomedical Informatics and Data Science graduate training program. I'm Paul Nagy. It's a pleasure to have you here today. It's really my great honor to have a colleague and friend coming today to present our Grand Rounds. We have Dr. Joel Saltz coming from Stony Brook. Joel received his MD in computer science degree, PhD in computer science from the medical scientist training program at Duke University. And then he went and came and joined us, and he was trained in clinical pathology at Johns Hopkins Medical School and served on Johns Hopkins faculty as professor and director of pathology informatics. He's truly a pioneer. He has launched new departments of biomedical informatics at Stony Brook, Emory, and Ohio State, and has served on the computer science faculty at Yale and the University of Maryland College Park. So Joel is a pioneer in developing digital pathology tools, methods, and algorithms with the ultimate goal of extracting and leveraging digitized pathology information to better predict cancer outcome and to steer cancer therapy. He is also an expert in high-end computing and has developed a variety of highly cited systems, software, and software methods. His research in pathology spans 20 years and consists of closely coordinated efforts in image analysis, machine learning, database design, and high-end computing. He has developed tools and methods through the years of funded projects supported by a wide range of institutions and agencies, including NCI, NLM, NIBIB, NSF, DARPA, NASA, DoD, and the DOE. His seminal work in digital imaging laid the foundation for digital pathology as it is today. He was the first to develop the virtual microscope and pioneered developments in digital pathology, whole slide image navigation, data management, and computer-aided classification. It's exciting to see Dr. Saltz's initial efforts included development of the first whole slide image viewer and devising efficient methods for management, caching, and supporting analytics carried out on whole slide data sets. This work has become the foundation of the new field of pathology imaging informatics today. And investigators, he mentioned, can be found carrying out exciting research in institutions across the country. So Joel, thank you so much for coming today and talking to us about how we can bring multimodal data together to create exciting new insights.
Well, Paul, thanks very much. And it's always a honor and delight to be back at Hopkins, however, virtually. I should say, and thank you for the very kind introduction. I should say Angelo DiMarzo and Bob Miller. I say Bob's on the call. Were a couple of my partners in crime at Hopkins in developing the whole slide imaging effort. And I have to say that I learned whatever I know about pathology, informatics, and systems from Bob in particular. So I have very, very fond memories.
And we've been collaborating since like 2004, but we have, but you had left Hopkins before I had joined. And so we've crossed paths in the night.
Yes, for many years. So, and it looks like things are heating up with our collaboration, which is great. And of course, Harold Lehman is another partner in informatics crime who I've known for many years and worked with recently at N3C and other contexts. So it's great to be amongst friends. And so anyway, so I'm going to give a talk mostly focusing on pathology informatics, but with the translational sort of clinical informatics spin, and then say a little bit about radiology, but mostly about pathology. Okay, so disclosure policies. I don't have disclosures, and this is the chat that one displays, which you've, I think, seen already in the introduction. Presentation objectives. No conflicts. I should say parenthetically, actually, when we developed the first whole slide imaging software prototype, which was actually leveraged some work that was done in a National Science Foundation Grand Challenge project at Maryland College Park, and Maryland people Alan Sussman in particular were highly involved in this. We went to the IP people, had a little chat. Fred Sanfilippo set us up with some venture capitalists, had a little chat. It was generally agreed that trying to patent such a thing would be a waste of time, and money would go into it. Nothing would ever come out of it. It was sort of an adorable academic curiosity. In any event, you can't patent software. So we didn't patent it, which is, I don't think we would have made a nickel off of it, but it would have been nice bragging rights. But in any event, that's neither here nor there.
Yeah, but okay. the world is better for it. How's
that? Well, such patents don't really stick anyway. I mean, they'd be kind of mean. When I remember there were a couple of patents in telepathology. Everybody was annoyed. I don't want to get into names even now, but everybody was annoyed. It's like, oh, they patented, now we can't do it anymore, and oh, they're so uncommunity spirited. And it's like, okay, whatever. This is so anyway, but certainly financial acumen at the individual faculty level is not one of Hopkins' strengths. It's still not one of mine personally either. Okay, so pathomics and clinical research. So in the last 25 odd years since I got into the digital pathology business, the whole context really has gradually but inexorably changed to a scenario where it is now possible. It used to be a promise, but it is now really possible to break diagnostic whole slide images that are collected routinely on millions of patients, classify them and segment them, tumor, lymphocytes. You can label the cells, you can segment the cells, you can characterize spatial distributions and morphology of glands, get collagen orientation. And when I gave the equivalent of this talk maybe 10 or 15 years ago, it was sort of more future tense, that you could take a region of interest or a single whole slide image or even a small collection. And if you had enough undergrads or grad students to edit the results of the then best of breed computer vision methods, you could actually get a pretty good classification of cells. We did a paper in 2011, 2012 involving sort of cell resolution based classification of GBMs, demonstrated the still useless observation that you can find small but statistically differences in outcome by cell characteristics. But that sort of work then took really a tremendous amount of manual effort. The algorithms were automated. You did get tens of thousands, hundreds of thousands, sometimes millions of segmented cells, but curating them and getting any kind of reasonable answer was a few hundred slides, and doing a semi-reasonable job was like a big reach. So the newer methods really are mostly about scaling, which is in truth much of the story in AI. So now quality control and validation is as important as it ever was, but you can now look at much larger patient populations with much more reliability. And then the notion is that you've got a bunch of Lego equivalents and you can build different biomarkers. So rather than sticking with the ones that the pathologist from late 1800s, early 1900s came up with and continue to iterate on, you can break images up into multiple scales of cells and ducts and crypts, as well as sort of more anonymous patches of various sorts, and you have a much larger space in which you can explore development of new ways of predicting outcome and really critically response to treatment. So this is I think both potentially a renaissance in pathology, although from a clinical AP point of view, it's really hardly even begun, but the technology certainly is developing, and also a major area of research interest. And then I make the analogy, which has been made many times before, that just as in flow cytometry, you sort of interrogate, count, graph, generally deal with populations of cells, you now have a spatial version of this in whole slide image analysis. So the area where I think this has become the most practically useful, although practical usefulness is really found more in cooperative research groups and clinical research studies, rather than on the front lines of care at this point, is in tumor infiltrating lymphocyte characterization. So TILs have a few advantages. First thing is, if you don't have a lot of TILs and you have a tumor, you are likely not to do that well. So they have personal advantages if you personally have TILs in most cases. But from a biomedical clinical point of view, because of the increased use of checkpoint inhibitors, immunotherapy, then TILs seem to be, in many cases, quite a good predictor. Lymphocyte infiltration tends to be a good predictor of outcome and increasingly response to treatment. And this is unrelated to informatics, but and then from an informatics point of view, they have the advantage, at least if you're looking at H&E images, which if you're doing large clinical studies, that's typically what you have access to, of being, little purple blobs on varying backgrounds. So the computational task is, relatively tractable, because you're trying to find the same kind of thing in a lot of different contexts. So algorithms for doing that, which we were one of the early groups to, publish validated algorithms in this. We did some work with the Pan-Cancer Atlas Consortium in like 2017, 2018, and related TIL patterns to molecular characterization in TCGA patients. So algorithms that do this sort of thing have been out for 5 years. So they become, I think, pretty robust. I mean, we're on our third or fourth, depending how you count it, generation of algorithms that do TIL characterization, and there are lots of other groups that that have these now. It's frankly, as these things go, not as hard a problem as many others. So what you're seeing here is, if you look at panel D, the yellow and red thing, what you have there is the result of an algorithm that classifies tumor being yellow and TILs being red. And these are 50 micron patches. And there are tens of thousands of these things in a whole slide image. So you're seeing essentially a thumbnail of a whole slide image classified as to where the TILs are. And so you end up, and then the off diagonal B and C are probability maps of tumors and TILs, and the top thing is the thumbnail of the H&E image. So you can see, quite visibly their patterns of TILs, and to oversimplify, at least in many cases, if you have large deserts where there aren't a lot of TILs, or there are very few TILs in the whole tumor, that's typically not a great sign. And some of the work I'll talk about involves looking at clinical studies and clinical populations with different types of tumors to assess this. And then E, F, and G are different types of cells, tumors and TILs at the individual cell resolution level. And I think this was done with an algorithm called HoverNet that the PathLake folks in the UK developed. I'll talk about some of our own algorithms that do cell detection as well. But the point being is that this is sort of gotten to the point where this is, we can do this on a reasonably industrial scale and have a consortium of NCI Seer sites where we're doing these studies, as well as working with ECOG Akron and various institutions. I should also give a shout out to Alex Soleil and his group at Hopkins. You probably know him, and his work involves a much higher resolution, very nuanced multiplex immunofluorescent analysis of TILs and response to treatment, where he and his group are really diving into the issue of predicting response to treatment. Mostly what I'm talking about here, I will talk about some multiplex IHC work. The focus of a lot of what we do is on dealing with large populations with data that is routinely gathered clinically, and both are very valuable. In one case, obviously, you have much more insight about mechanisms in much higher resolution, but on the other hand, it's very expensive and very time consuming to do the studies. On the other hand, if you have a large population, real world data or a clinical study that closed 10, 20 years ago, 5 to 20, whenever, you basically, as long as they didn't throw out the glass slides, which knock on wood, they never do, you get them scanned in, and then you can do this analysis. So you don't need to rely on the study having been set up front to do this stuff, or availability of tissue blocks. So I mentioned, so we collaborate with various different groups. I'm part of the data science AI sort of committee at ECOG Akron, trying to sort of move things forward with this. One of the projects that we launched like 4 or 5 years ago, we put in a proposal to call into an RFP for the Seer program. And the notion was that, can you augment Seer data? So for people who don't know, Seer is a NCI program that sucks in and curates data for a third to 40% of American cancer patients. And so there's a whole pipeline that comes from Seer states, hospitals forward pathology reports, and other correlative data, and the Seer folks, huge amount of manual effort and some actual AI now do curation of this data and then generate large data sets. So if you read studies about breast cancer of type X is 3 times more common than type Y, or is 20% more prevalent than it was 30 years ago, or whatever it is, these things often come from Seer sites. So the idea is that one should be able to leverage additional types of data to essentially take advantage of Seer's data sets as real world research data, or at least real world nuanced surveillance data. And so we said, okay, we would add digital pathology to Seer data. And at the time, it was a bit of a wing and a prayer, because we were just going through the process of improving algorithms and going from engineered features to deep learning methods in, say, 2015, 2017, when we were getting this thing funded. So it was a little bit of a wild and crazy thing to say, well, you've got millions and millions of slides, you're going to automate that. Well, so now it's a much more reasonable thing. And so we have a consortium. It's a mini consortium. They're just 4 Seers. And we've gone through all of the fun stuff that, analogous, Harold, to, the N3C, although it's obviously much smaller, with fun things having to do with IRBs and permission and all of that. So it takes years to get these things sorted out. So we're finally up and running, which is nice. So the notion there is that you should be able to get highly curated clinical characteristics and then relate that to routinely acquired results from routinely required pathology data, TIL maps, and tumor infiltrating lymphocyte characterizations in their predictive power, sort of a main feature, but we also have a project involving pancreatic subtyping and colorectal budding. So I'd mentioned the work we did with the Pan Cancer Atlas group five years ago, involving about 5,000 cancer patients, 13 cancer types. And it was the first of many of our studies that looked at spatial TILs as relates to outcome. A bunch of other people are doing that as well now. This slide shows some of the work that we did. Arvind Rao was the lead in this. Arvind is now at University of Michigan. He's a biostatistician with specialty in spatial analytics. The whole issue of, well, okay, if you've got pathologists, and pathologists aren't going to look at every little 50 micron patch, and if you randomly sample little patches and you're even 5,000 images, you've got billions of patches. So how are you going to get a representative enough sample to do quality control? So one of the things that he did was to develop analyses, and these are based on standard statistical methods that were applied in other areas, involving relating human observations of larger regions that were qualitative, low, medium, high TIL, to model-based observations of the same regions, and then looking at the statistical characterization of these things. Another thing that one tries to do, at least periodically, is to relate the data to molecular data and data from other modalities. So in this case, there is CyberSort that we use to do comparisons. You know, now no one would probably do more IHC as well. So we did a bunch of work involving parameterized characterization of TIL distributions. So on the right are the qualitative characterizations that Alex Lazar, who is a sort of lead pathologist from MD Anderson on the project, adapted from his melanoma work. And Arvind Rao came up with basically a bunch of spatial characterizations that were used in ecology and things like that, which frankly didn't necessarily have a lot of intuitive feeling for most of us, but it defined essentially the density of clusters, the type of clusters, the dispersion of clusters, and things like that. And you can see, so this is an example down below. You have sort of a TIL desert. You're not seeing the tumor there with lymphocytes surrounding the tumor but not getting in. And sort of teleologically, that's generally a bad sign, in the sense that the tumors are, that the tumor is protected from the TILs, or at least they're causing them to feel senescent and unenthusiastic. And I should say, you only have to go to the Alex Soleil project and the biological people in that group, and they're real experts on the whole mechanism. I heard a bunch of talks with them recently as to how all of this happens. And then above, you've got a tumor with lots of TILs, spread out. And then so in the cell reports, in the cell immunology papers coming out of this, there were a bunch of analyses involving different tumor types and different impacts of different clustering. So more recently, we've taken a much deeper dive into looking at this in particular cancer types, because the sort of overall, view of everything, you can say, well, yeah, TILs are probably on the whole better than no TILs, but it's very high level. And so we've worked with Melissa Trauester and her group from UNC, and after doing a bunch of work with TCGA, and they actually were the curators of a lot of the TCGA breast cancer work as well. We then sort of replicated some of the work we were doing on their UNC breast cancer study, sort of breaking things down by race, stage, grade, and basically looking at the sort of characterizing the impact of TILs, high versus low, for instance, on different molecular subtypes, and looked at hazard ratios, and then went on and characterized the impact of different patterns of TILs on outcome, and found that there, as we had hypothesized, there were sort of risky patterns of till deserts, and lack of till forests, and in a broader sense, actually, and Raj Gupta and his folks have really been taking the lead in this in our group, essentially characterizing tissue type maps and till maps for different types of cancer, and then carrying out analyses to sort of make sure that the classifications made some sort of sense. So this is actually, and again, from a clinical informatics point of view, I think that we've really, at least the way we handle much of this work, we actually have a group in computer science. Dimitris Samaras is probably the lead person in the group, but some of our biomedical informatics people, Chao Chen in particular, are very, very algorithmic. And I'll talk about some of the algorithms. And then we have sort of over in biomedical informatics, much more of a focus on, okay, we have these tools, we validated these tools, or you always have to revalidate. What can you actually, do with them? And how do we sort of aggregate reasonable patient populations, integrate different types of clinical data, work with cooperative groups and real world data groups to actually use this stuff. So it's kind of a translational sort of thing. So that's a real high level description of some of the TIL work. And I'm going to give say a few things about some of the algorithm work. And I think we've got a good connection between the folks who actually need to use and develop this stuff and the people who develop the algorithms. We, write papers together and meet together actually many times a week. So this is a wonderful student of ours, Shahira Abusama, who has been doing work in a number of areas, and in particular, classification of cells. And so one of the things is when you're getting beyond TIL classification and looking at some of the more nuanced, multi-class cell classification sorts of things, one of the things that tends to be very tricky is that even with the sort of better of the current algorithms, and HoverNet really is as I'd mentioned before, is sort of a state of the art. You have a lot of annotation to gather and a lot of validation and revalidation to do, and you can have pretty high error rates in tissue that has not been evaluated before. So new algorithms can be useful in this, and one of the things that she and Chao Chen, one of the faculty in my department, who I actually recruited to do radiology, but I keep on getting people involved in pathology. He's a topology guy. And so basically the types of algorithms that he develops are sort of a cells of a feather flock together sorts of algorithms, where you basically have loss functions in your deep learning algorithms, which take into account spatial clustering and take into account topology of various different sorts. So this one uses a Ripley's K function, or a modified Ripley's K function, that captures sort of local density of different types of cells, and then uses that in a sort of conditional probability implicitly context. Harold would be pleased, I guess, that there's Bayes in there someplace, to improve the ability to predict, and it definitely does seem to be helpful. one of the things that is probably most important, and the reason why it kind of got developed, was developing detection. Basically, if you can dot nuclei or even give statistics as there are 37 of this and 81 of that within a patch, that's much weaker training data, much easier and faster to generate than having to electronically circle every miserable nucleus, which can take a long time. And we had a bunch of undergrads doing that sort of thing. And interestingly, Alex Soleil and his group have a bunch of undergrads doing that sort of thing. It's pretty painful. And so weak training is very useful for these sorts of things. And so an approach where you take a region of interest and you count or estimate, but you can even count, you can just count them. You get a number of different types of nuclei, but without having to sweat the details of where exactly everything is or what the boundaries are. So she's published a couple of papers on this, and in her hands, it does a little better than HoverNet, but I suppose every student's algorithm does better than everything else because that's their thesis. But it certainly does as well. I think I'm pretty confident of that, and seems a lot easier to train. So, and that's motivated not so much by the love of new algorithms, but by the dislike of training. And I think a lot of our work, has that theme, that the more computer science algorithm focused people come to us and say, well, what should I work on? And then we have a negotiation. I say, well, we need this, this, and this. And they say, well, this is boring. I don't want to work on this. And then sometimes I try to spice it up a little bit, but sometimes they don't bite. But then that might be interesting. I might be willing to go for that. So then this is another one of Shahira's methods. Mamadou Hassan, Chao Chen have also contributed to this. So this is analysis of cell populations in multiplex immunofluorescence. So it sounds boring, but maybe it is, I don't know. But these are whole slide images, and our particular machine is a Roche Discover platform or whatever, and it just basically paints, has markers, 6 to 8 markers that give you different molecular information, and you scan this in with a Leica or Olympus. It's a whole slide scanner, not a multispectral scanner. So they're cheap whole slide scanners, fast. You get RGB at each point, and then the goal is to say where all the cells are. And I felt a little sheepish as to how long this thing took for us to develop. We published a paper or two, a couple of papers on it, and it works. But then I was pleased actually that Alex Soleil is tackling a different problem involving multiplex immunofluorescence. And he has a whole bunch of, he said, we do whole slide images unlike patches. Well, we do too. And it takes him a lot of time too. So but we have it pretty well automated. And so once one does this sort of thing, then you want, the various driving problems that lead one to this. A particular collaborator we have that got us into this is Ken Schroyer, who's the pathology chair at Stony Brook, who's interested in a marker called keratin 17 and its impact on pancreatic adenoductal carcinoma outcome. And this has been kind of his research passion for a number of years. And so, but the questions he wanted to answer in grants funded by the Pancreatic Cancer Foundation in particular, involved the tumor microenvironment. So what is the impact of keratin 17 on neighboring immune cells? And this is actually an oddly difficult thing to get a hold of. And one that required a little bit of battling with the computer science people because they didn't find this interesting enough. But we persevered. And so the issue there is you have a whole slide image in pancreatic cancer. So if you look at the brown and the teal, they're both pancreatic cancer, the brown being keratin 17 positive, the teal being negative. And so you have like all these weird boundaries. It's kind of a fractalish kind of a thing. And then you want to say, okay, what is the impact of having the brown stain versus the teal stain? I mean, obviously not literally the stain, the different type of cancer on the tumor microenvironment. And so we went through a number of iterations, is about a year, not full time, obviously, but the grad students spent for a lot of time on actually characterizing this. And ultimately, we're able to essentially generate data which looked at the relative tendency for, say, CD8 cells to be close by boundaries of tumor that had keratin 17 versus boundaries of tumors that were not stained by keratin 17. So, and then to look at the densities in keratin 17 positive versus negative zone. So again, the reason why one would want to do this is, at least the reason why our collaborators wanted to do this, is that they had a marker that worked. And then they wanted to say, okay, what does this have to do? Why do we think it works? What's different about tumor that has this marker versus not? And it turns out, and I'll mention this in a little bit, there's the classical versus basal distinction in pancreatic cancer, which ends up being more or less the same distinction. So then the question is a little more, is a little broader than this particular investigator's question about keratin 17. The two now acknowledged subtypes of PDAC are classical and basal. Basal has even worse prognosis than classical, significantly so. And what's the actual difference between them from a microenvironment immune point of view? So that methodology is useful in this. So now I'm going to switch gears a little bit and talk about some work we're doing as part of a broad consortium with the FDA. And this also involves tumor infiltrating lymphocytes by semi-coincidence. So Brandon Gallus from the FDA has put together this broad set of consortia, industry, academic, with the goal of developing a mechanism, sort of templates for validation of AI algorithms in pathology. So the idea is, and he's a statistician, but he's been working in imaging for a number of years. The idea is that you end up having a hidden data sets along with analysis methods that would be used, essentially analogous to like a challenge. And somebody who says, yes, I have an AI algorithm would say, okay, I'd like to test my AI algorithm on your data set. They'd get permission to do so. They get a grade, and then they could use that grade when they do their FDA proposal. So the idea is that the FDA kind of pre-cooks some exams for aspirants who claim they have an AI algorithm. And that way the FDA and the people applying don't have to go through the, well, this is how we tested it. Well, what was your methodology? I don't believe it. How did you know? How did you test? How did you validate? And so forth. So it seemed like a really good idea. And when he was getting into this, we were getting into TILs, and Roberto Salgado, who's a very dramatic fellow, who's a pathologist who's into TILs, but manual TILs back then. So he was like a big proponent of TILs. And so basically, we got together and decided that TILs would be the test. And our flavor of TILs just asks the question, you have a patch of tissue where there are more than 2 TILs. His method is much more nuanced and involves looking at stromal areas outside the tumor, but not too far away and not necrotic. Long set of rules, a lot of training needed. So it's a somewhat nuanced task. So this has led to a bunch of studies, lots of papers, and nothing if not a gradual approach. So the pre-study after 3 years is done, is published. basically how you choose the ROIs, how you rank the ROIs. Pathologists were recruited from USCAP as subjects, data gathered. So now the group is launching on the official study, which would then lead to a prototype, put it away and use it as an AI validation algorithm. But I think it's very exciting in the sense that if any of these things are going to be, reduced to clinical practice, they have to be approved. And to the extent that the approval process is transparent and not horrible, then they can be done by larger sets of people. And in fact, if I were going to develop lab developed test and get approval, short of full FDA approval, if the FDA had something like this, I imagine I'd probably use it. I mean, so I think that, basically the more that one can get sort of well accepted methodologies and referees for doing validation, the more acceptance these tests are going to get, as well as you do want to know if they're not going to work. So you don't want to be fooling yourself. So the powers that be have to agree, whether it's an IRB or whether it's a state or whether it's CAP or whatever, that these things are okay. So he generates all sorts of statistics involving interhuman observer variability in pathologists. Visualization. And I should say also from a clinical informatics perspective, so C. Wen, who's listed as one of the authors, was a PhD student in our group who then went to the FDA, and Victor Garcia's was a clinical informatics, ACGME fellow who's also working for the FDA. So our program has actually fed a couple of people to that group. So now a few words about data management and curation. So this is as Paul mentioned, how we got into the business in the first place, which was visualization, data management, so forth. And so this lives on in the Cancer Imaging Archive. The great grand browser coming from the virtual microscope at Hopkins is used in TCIA if you want to look at whole slide images in TCIA. And then we've been developing a bunch of other infrastructure. For people who don't know, TCIA is a big deal. Fred Pryor is the PI. I'm an MPI kind of for pathology for the ITCR funding of this, but he runs an amazing operation with zillions of users and things like that. And so our group has developed also the database infrastructure for managing whole slide images and features. So bulk data annotations, if you want to, if you have an algorithm that detects or segments like every nucleus and every whole slide image in a data set of thousands or hundreds of thousands of whole slide images, you end up with billions to trillions of little polygons. So there's quite a data management thing to be done with that. We and most people began representing these things using GeoJSON. There's a DICOM group, Working Group 26, Supplement 222, as Paul knows well, which has come out recently with maybe 6 months ago, describing how bulk data annotations, things like segmented nuclei, should be represented in DICOM. So I think we're all gradually moving to this, but that's just the first version because it's kind of simplified, but it's a wonderful thing as a start. So in the context of imaging DICOM, when we talk about imaging DICOM, management and representation of annotations, in my view, are extremely important. For instance, if you have a prostate cancer excision or biopsy, and you have different Gleason grade regions segmented, which are in other people's algorithms do increasingly well, and at least in a clinical research setting, I think will become sort of a standard thing to do. I mean, we're working with the Miami group looking at TILs in an active surveillance project, and actually, come to think of it, I should be talking to Angelo again here about work that we've talked about kind of along those lines. But in any event, so I think actually representing annotations of regions in cells is going to be very important in a research context, and then metadata that summarizes this. Say, okay, 14% Gleason 4, or 18% non-small cell adenoleupidic, or something like that, which can actually be derived from the annotation. It doesn't have to necessarily come from the pathologist, and may well not, because the pathologist may not describe things at that level of detail. So some of the new browser work we've done, Eric Bremer in particular, this has been this person's grad student and a staff's passion for the last couple of years. He's developed some very sort of nuanced software that allows one to look at multiple windows and label things in different ways, and essentially take bulk data annotations and represent them overlaid on top of each other or next to each other. So some of those maps I showed you earlier, so the use case, the reason why I pushed him to do this, actually, he was happily doing some other project as kind of a side thing for his daughter involving graphics, is like, if you basically say have a bunch of several different neural networks that supposedly calculate the same thing, or we were looking at a federated learning algorithm with Spiros Bacchus at Penn versus our non-federated algorithm. Are they getting the same answer? If it's saying like 37% versus 28% tills, it's like, okay, that's nice. But you want to see the probability maps. You want to see, what gets labeled, what doesn't. In many cases, what gets labeled as tumor is in the eye of the beholder. So there's a lot of tumor-associated stroma that some models call tumor, some don't, and that can be important in some context. So ability to actually get in there and look at different things. We actually had one of my MSTP students developed a highly optimized version of some predictive models we had, and the results were a little bit different. Not much different, but they shouldn't have been different, at least hardly at all, because they were predictions. And to make a long story short, getting down at this level of detail let us find areas where there were little blocks where there was significant difference, and he was able to actually debug the algorithm. And it wasn't anything that made it absolutely horribly wrong, but it was a small error, and it would have been really hard to sort out without essentially using something like this as a debugging tool. So this is his software. This is something that's now open source. And what this thing actually does is a little bit different. He has one software system that does the highly optimized prediction, and it's about 10-15 times faster than the old version. So and it's publicly available. This model is actually designed to evaluate different models on a standard test data set. And these are specifically patch-based models. So a bunch of tasks involving tumor classification, TIL classification, and set up so that you can go to model repositories and look at different pre-training methods and different hyperparameters, and very easily set up comparisons. Okay, and the reason why we did this is our computer science algorithm developer, wing of our group, was always doing comparisons with state-of-the-art algorithms. I mean, everybody does, except when you do comparison with state-of-the-art algorithms and it's the graduate student going to GitHub and downloading and interpreting the state-of-the-art algorithm and putting in what they think are the right hyperparameters. Almost none of these, almost very few published works are, have enough reproducibility in terms of the being able to do exactly what was done in the paper. there's generally the code's available, sometimes the model's available, sometimes the data's available, but rarely is it all put together in a complete enough way that you really can say if they got an AUC of 0.893, that you would get an AUC of 0.893. So, and I think that's a real lack in the community. And so we wanted it for ourselves. And so this is also available and it's out to be published, submitted. So finally, a few words about modeling human attention. So this is really a different project from the other ones I've talked about. In its motivation is to emulate what to understand and evaluate what pathologists do. So specifically, so we have a cognitive psychologist, Greg Zielinski, different one, and Dimitris Samaras, myself, and Raj Gupta, and actually growing number of students, but the most senior of whom is Surjeet Chakrabarty. And basically the notion is that we are developing models that emulate what pathologists do. So attention patterns of pathologists. So when a pathologist looks around in a slide, can we produce a artificial intelligence pseudo-pathologist that doesn't just come up with the right answer, but anticipates essentially what the pathologist would do or what the pathologist would do on slides that they haven't seen yet. And the notion there is that if we understand how pathologists go about doing things, we can essentially look at patterns that tend to be more correlated with good results than others. We can characterize expert versus versus novice, which a bunch of people, Rebecca Crowley, by the way, I want to give a shout out to her from Pittsburgh, who did a lot of the very early work on this kind of thing, but she didn't really have AI models at the time. So I think we're sort of doing, for people who are familiar with her work, that sort of thing. We're using, of course, our whole slide image browsers that are instrumented. And then we essentially aggregate scan paths. So in the initial set of papers on this predicted the attention heat maps. And these were quite small data sets with like 50 to 75 pathologists looking at a little data collection of 16 whole slide images. And we're launching a much bigger study. We got funded through the NSF, and we have a pending NIH proposal and got IRB approval to do a much bigger study. So we're about to launch that. So basically you look at where in the slide people look, what power they look, what the sequence is, initially predicting what is going to be looked at the most. And one of the things that you see, which is not surprising, is if you look at, say, the visual attention heat map, and this was the task was to tell us what the Gleason grade of a whole slide image was, that what they tend to look at is the tumor region. So the non-pathologists found this to be an amazing magical thing. It was like, okay, what else are they going to look at? Okay, so I view this as a little bit more of a sanity check. But in any event, so there have been a few models that classify a degree of attention in patches, and some of the new work that I'm really excited about uses some transformer methods to actually predict the scan path, which obviously there's a lot of nuance in terms of predicting scan path that we could get into if people are interested, but I don't want to go over. So I'll leave it to say that there's a lot of inherent uncertainty in the world. So predicting the scan path doesn't mean that, it's going to say you're going to CVS tomorrow at 1. I mean, it's the sort of scan path a person would do rather than and I pointed that out actually to the grad student, and it was like, oh, that's it. I mean, it's interesting. These multidisciplinary things have a lot of interesting things. In any event, so it's work in progress. It seems to be getting a good reception, from the journals, but and the goal is really to have a method that, as people move to digital sign out, would say, well, this is interesting. You're going about this way. Well, maybe you should look at that. I mean, I think you don't, I mean, there's a whole human factors stuff. We don't want everybody to hate us, but this is this is sort of basic research to sort of see if we can understand how to do this. The how you build it into a decision support system without creating a legal nightmare or having the pathologist kill you is probably somebody else's problem. But I think being able to have a computational pathologist avatar would seem like a cool thing. Finally, just one last radiology example. We do have a bunch of radiology work that has a lot of overlap with both people and topic. We were heavily involved, as Harold knows, in the N3C thing, which didn't in particular involve imaging, but we were sort of the cousins, the imaging cousins, and we were involved with Midrick, which was imaging. But one of the cool things, this was a Chao Chen thing that just got published, which involved, we have an aortic aneurysm project with the head of head of surgery actually now. And the notion is to be able to assess rate of growth of aortic aneurysm based on imaging and clinical data. So one of the things that we ended up wanting to do was to get a very high quality segmentation of the aorta, in addition to the sort of less supervised versions, of this. And so this led to some very cool work, which involved using topological constraints about body organs in deep learning algorithms that do segmentation. So this is, I think, another example. And in many cases, these projects actually lead to interesting technical papers, which you could see how it fits into the big project. Our success in the big project to date has actually been very much clinical informatics. We now we use NLP, nothing special, Bert, to feed incidental findings to the clinic, so they follow them. So they're following a bunch of people they wouldn't have followed otherwise. So there's a big separation between the practical stuff we do, which makes the head of surgery like us, and then the more theoretical stuff, which I don't know if the head of surgery really likes or not, but the computer scientists like it. Gets published good places, and it might help actually with the task. In any event, okay, so I think I'll wrap it up. There are a bunch of people involved in this, but I think it's really our core faculty. Actually, most of them are in the BMI department. Dimitris Samaras is in computer science, but pretty much everybody has joint appointments in computer science, and we have a lot of collaborators there, and lots of consortial teams of various different sorts, and a bunch of grants to thank, and happy to take questions.
Joel, thank you so much for that presentation. I was thinking, do you see, I was trying to understand the differences between active learning in radiology versus pathology, in terms of how, to mimicking an annotator, how does the resolution play a role? The multi-resolutional challenges play a role with creating, active learning training sets. Do you see a difference in those two approaches?
Oh, okay. So I'm sorry. So I don't know if I quite understood your question.
With pathology, you have a resolution problem. You've got many tiers of resolution, where in radiology, we often don't. And so when we do active learning, we're basically trying to do segmentation tasks at scale. So we're able to have a fairly good, so we've seen some pretty successful active learning models for helping with annotation. Are you seeing similar productivity with automatic or active learning annotation in pathology, or are there additional challenges with the resolution?
No, I mean, okay. So we actually, I didn't mention it, but we have an active learning effort, published a couple papers with a group in Brazil who used one of the head of the group, George Teodoro, used to be a fellow of mine. And so we do have an active learning, and it seems to be very promising, actually. We're just kind of in the transition of actually beginning to use it. It makes a huge difference, because even at a single resolution, if you're looking at tumors or TILs or cells or whatever it is, there tends to be so much kind of area. These images are very large that hunting out new examples, if you do it the old-fashioned way, and you're not doing well on, Gleason 4 or 5, and then you got to find regions that look kind of different than the regions. You basically do a prediction and see where the prediction doesn't match, and then you take it. So whereas if you have a more dynamic approach that analyzes, this kind of on the fly, and so one approach is kind of crude. We have sort of stripped down neural networks that kind of gather up patches and say, here are the ones you should look at. Chao Chen and a student of his have a more sort of systematic estimate uncertainty and likely error. But I think one of the things that's really exciting in multi-resolution, to answer your question, and I didn't get into this, but so as a bunch of weak learning and essentially multi-instance learning methods where you have like high versus low till for a whole slide image. If you train using an algorithm that uses that, and the one we're using now is something called CLAM from the Mahmoud lab, you basically can look at the attention map, and it recapitulates the map that you would get by using little patches remarkably well. It's not perfectly, because what it is it answers the question, how important was this little piece of tissue in making the decision? It doesn't actually say where the tills are, but it's a good hint. And so integrating that sort of weak learning into a strategy. The other thing is, and this is something that Shahir is working on now, is basically dealing with the multi-scale stuff. I talked a little bit about how our current algorithm, uses sort of summary information from a region, so you can extend that. So active learning is useful in pathology already, and I think multi-scale active learning is a bunch of people are working on. I mean, they may not call it active learning, but at the end of the day, the interest in weakly supervised methods and self-supervised methods for that matter. We have actually a student who is using cellularity, basically self-supervised method, to pre-train a model to recognize cellularity. So there are just too numerous to count different wedges into this. So I wouldn't call it a problem. I mean, it's kind of an opportunity. So so there are a couple of questions. So tension density study, do pathologists study think that they are real patients and real reads? No. The IRB wouldn't let us. And besides, I mean, these are, at the moment, these are TCGA images. And, we have to submit to our IRB what we're going to say. And, this is the drill, mother may I. And certainly I mean, yeah, so no, they know. And it does concern us a little bit. One of the things I have a resident, we're beginning to get residents, I'm sure, as you with IT-ish backgrounds. And so one of the things I want to do is actually instrument our Apereo viewer. We're rolling out digital sign out in the next few months, and compare what essentially pathologist behavior with a real viewer and real sign out on our instrument, because that actually does concern us that these guys are not taking it seriously. So
when I was at University of Maryland, we did get an IRB once to study the process of critical findings, where we were able to inject a fake case into the real packs of a critical finding. And so it was an enormous IRB undertaking to do this. And we had to consent the entire department that you may or may not be getting cases.
Right. I mean, somebody might become, that's funny.
Yeah, it was, and you have to, of course, creating a fake patient in a real patient MRM database is a pain. And then we ended up having the spouse of the PI being the one that you would call as the referring physician for any critical finding. But it was a big to do.
Yeah, but I think with this sort of thing, if we actually, if we are able to instrument during actual sign out, I guess we'd have to have the IRB. I mean, this is really just in the talk phase right now because we haven't rolled the thing out. But if basically the IRB doesn't object to us putting some hooks into the Apereo viewer to track what's looked at, assuming they're not there already, we frankly haven't played a lot with it from a software point of view, then we can see what the pathologists really do for sign out. And we already have permission to reuse slides in various different research studies. And so if we de-identify it, we already have a protocol, for that. And then we could use that as one of our slides in a de-identified form and get, so the sign out, the real sign out would be a real sign out. And then the fake sign outs would be known to be fake sign outs after the image had been captured.
Great. We are over time, but I wanted to thank you again for giving that great grand rounds. And we really appreciate you with a great lecture on pathology informatics. That was exciting to see.
Okay, well, thanks for inviting me, and it's always good to be at Hopkins virtually. Thanks.